Feel overwhelmed when you read research papers? Think research is being conducted in labs far, far away? Well, tune into the AlmaMAC Thursdays from 12 to 12:30 p.m. where we interview McMaster graduate students about their research. You learn about important research that's happening right on campus. Learn about what the guests did before research, how they got involved in academia, and what kind of impact their research can have on you. The AlmaMAC is covering it all from Thursday 12 to 12:30 p.m. on 93.3 CFMU, redefining radio in your community.
Hello and welcome to the AlmaMAC. I am your host and CRISPR baby, Adam, and today whoa, that's not good. No momentum. How's this? Does this work better?
No.
Okay, maybe jump on mic 2 then, which is this guy over here.
Testing.
Are you good?
Yeah.
Try one more time.
Boom, 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 boom. The buttons are on. Oh.
Okay, so that's very quiet. All right, well, regardless, we're going to make do. Hi, I'm Adam, and today I'm with Sean.
Hello.
Oh, man, come on.
Oh, this is the new mic 1.
Yeah, so I got this one turned up here.
Okay. And then mic 2 is just not... Can we try mic 3 again? Ooh, no, that's not cute.
Why are we getting this howl? Maybe if we turn down the control room.
Do. Yeah. Okay, it's working. We did it.
We're in. All right, welcome to the show. We're the AlmaMAC, and in lieu of an interview, we have a news story we want to talk about.
Ooh.
So, on December 30th, 2019, Jiankiu was sentenced to 3 years in jail and approximately fined about $500,000 US by the Shenzhen Nanshan People's Court of China. So, he is an acclaimed scientist. He studies gene editing in Southern University of Science and Technology, which is in China.
Or studied, because he's in jail now.
Yeah. I mean, that must suck.
Yeah.
But we'll find out why soon. He led cutting-edge research on the enormously powerful CRISPR and Cas9 gene editing technique. So he picked this up during his postdoc, which he did in Stanford. Let's see. He was listed on Time Magazine's 100 Most Influential People of 2019, based on his highly praised announcement of creating the first genetically edited human babies, which he announced in 2018.
Yeah, at a conference.
Yeah.
Yeah.
So what's the deal with gene editing? Why is he doing this?
Well, he was trying to he was trying to confer HIV resistance to babies, essentially.
So that sounds like a great thing. Why is it?
It sounds great.
Mm-hmm.
But how it was done was questionable. And that's why he's in jail right now. So
I kind of remember this news story in 2018. It kind of stirred the pot a little bit. Some people thought that this was awesome, that, we're using this CRISPR technology, which is pretty much brand new. It's medically speaking.
Yeah. Well, bacteria have been using this system for years, not years, but forever. Yeah. So essentially, CRISPR is short for clusters of regularly interspaced short palindromic repeats, which is something you don't need to know. But essentially, it's a stretch of DNA nucleotides. And it's really CRISPR-Cas9. So people just say CRISPR, but really the technique is CRISPR-Cas9. And Cas9 is the enzyme that bacteria and some archaea use as a defense mechanism. So if a virus has approached a particular bacteria, the CRISPR-Cas9 system will essentially recognize that, like, the DNA from the virus, and it will be able to launch an attack on the virus, protecting the bacteria and the archaea. So what scientists have discovered, though, is that we can potentially use this system for like outside of bacteria. So in plants, for instance, we can use this CRISPR-Cas9 system to recognize particular DNA sequences in various genes, where we can edit the genomic landscape in these particular genes. So, for instance, in certain crops, it might be advantageous to remove particular genes that could be, for instance, like crop length, like how much crop you'll get after a particular gene, that will predispose the plant to producing more or being resistant to particular bacteria, viruses, any pathogens in general. So you can use this system to prevent pathogenic diseases.
So we've done a really good job. Well, not me personally, or you might have done some work on this. Maybe not. Humanity has done a really good job of like understanding DNA and what these things code for, is my understanding. And this Cas9 CRISPR technique is almost like a targeting system to like bring our knowledge and force it into little spots where we want it to go.
Yeah, essentially. So he inactivated a particular gene, CCR5, which is a receptor that allows for the HIV virus to invade healthy cells, essentially. And this caused a lot of outrage in the academic community because of how it was done, basically.
Okay.
Yeah.
So certainly whenever you, from the people we've interviewed, it seems like anytime you want to do some sort of medical testing, especially on humans, there's a lot of ethics board consultations, and that kind of makes sense. But in terms of this gene editing stuff, what kind of like things do you have to think about in terms of ethics? Like, what could be...
Even if you're not doing gene editing, if you're doing any type of research where you're going to manipulate an organism or take it out of its natural environment, you have to do some kind of ethics. So my lab, we work with mice. We have to do ethics. I specifically work with humans. I have to do ethics. And it's not like I'm actually taking these humans and like editing parts of their DNA. I can't imagine. Like what I'm doing for my work, I am taking tissues from particular breast cancer surgeries, and I'm doing experiments with these tissues. So I'm not working on the human directly, but I'm taking a part of these individuals and doing experiments on them. I need ethics to do that.
Yeah.
So, of course, you need some kind of review board. So, if you're listening and you're not familiar, you have to submit your application before you can start any of your experiments. You have to submit your application to an institutional review board, or IRB, and they would meet and they would basically have their list of criteria as to what is deemed right, or how far, quote unquote, how far you can go with your experiment, and how it would basically list out the list of things you need to do for how you enroll patients or participants into a particular study. You need to tell the participants XYZ, you need to let them know of any risk that may be involved, you have to let them know of any benefits also. You have to just be very transparent. So that is a process of going through ethical review, and it takes time. Sometimes it takes a lot of time, and every year you have to sit down and do this. You have to apply for the renewal for the mice, for the human work. Even if you're not working on mice, I read somewhere someone is working on sea urchins that don't have like a fully developed nervous system and can't necessarily feel pain, and you still need to go through ethical review for that.
Yeah, I heard if the thing doesn't have a spine, then it's a lot easier, but you still got to do it, right?
Yeah. How did he get away with doing this on babies?
Yeah, my understanding is a lot of paperwork, sleight of hand, a lot of misleading the people who are involved. Sometimes you hear in the news, like, these sort of things happen in different countries. Like, you have different ethical boards operating in different countries. So this happened in China, but it's encouraging to know that I think the people that are coming down the hardest on him are the Chinese government. They are not happy with this.
Yeah.
I mean, understandably, but it wasn't just happening in his country. He also has collaborators in the States.
Yeah.
And some of these people are also getting brought into this.
Mm-hmm.
So he has two Chinese collaborators who also got lighter sentences, but I know his PhD supervisor from Rice University is also coming under some scrutiny at this point.
Yeah.
Because, you know, you can't just have one person go into the lab and do all of this stuff.
You have to have some, like, the money has to come from somewhere to do your research. You have to have funding. You have to have, like, you're not just going to do this one big experiment all on your, like, by yourself. You have to have a team working on it. And that baffles me as well. Like, the people who are working along with this entire situation, how?
Yeah, so there's one sort of narrative that I've heard that sounds attractive until you sort of put it under a little bit more scrutiny of like the potential for, you know, a great breakthrough that, you know, helps a lot of people. If it's getting bogged down by all this bureaucracy and everything, then isn't this guy like the Batman of science? He's going in and doing some great work, even though it's ethically not so sound.
But it wasn't even... So the concept of what he was trying to achieve, I can understand, okay? As scientists, our goal is to bring new knowledge, right? Is to answer questions that haven't been asked before. To just be curious and, yeah, like just do things that people have never done before or explored before. And to bring about greater good, really. So I can understand the essence or the spirit of trying to tackle HIV, but not in this way. It was sloppily done.
Yeah, I also saw an interesting point. Science Sam's Instagram.
Hi, Sam.
Hi. There are ways to live a fully happy life with being HIV positive. This is
100%.
This isn't really. If you're going to go and do your Batman science, I feel like there are other things to do it on.
Yeah, because with like the advent of modern science as we know it, the antiretroviral therapies work quite well, and in most countries they're readily affordable. And for people who don't have HIV but want to prevent HIV, there are therapies such as PrEP that people can take every day. So, and those are also quite readily available to, at least in North America, and throughout other parts of the world. I know there's a huge push for this worldwide. So, like, this type of therapy would be a lot more expensive for people who cannot afford it. Like the whole IVF nature of it. That's expensive. And then adding a layer of CRISPR technology on top of that, it will not make it any cheaper. So
I don't want to be a conspiracy theorist or anything, but it almost sounds like there might have been other motives going on behind the scenes. And I guess we kind of have to wait to see how the investigation... I mean, the investigation is over, but what comes out of it to see what exactly happens. But something that I feel like we didn't touch on is that he was doing... It's going to mess me up every time because his name is he. He was doing all of this work, and it wasn't like ex vivo. It wasn't outside of the body. This went into real people, and they were birthed. They have code names to hide their identity, but these are two human beings that he tampered with. In the media, they call them Nana and Lula.
Yeah. God knows what's happening with them right now. Like, we have no idea, obviously, to protect their identity. We don't know. Yeah, we don't know. Because as far as I understand, this particular mutation, the CCR5 mutation, also also have the potential to cause other immune response to such as West Nile virus. So it could potentially cause the babies to be more susceptible to getting the West Nile virus. So, yeah, like, here you are trying to this is another reason why I think, like, the entire thing was just sloppily done. I don't think enough preliminary research was done to understand what would be the are off-target effects, essentially, of this particular mutation. For this particular disease, like you have to take into consideration what's going to happen if you alter the genome. If you alter the genome in this particular region, what's going to happen to the rest of the genome? Are you going to cause susceptibility to other diseases? Are there going to be other characteristic traits that could be, I don't know, like there's so many things you have to think about before you can just jump and say, okay, I'm just going to edit this genome. Yeah. Just like that.
Okay, we have a quick commercial break, but when we come back, we're going to maybe touch a little bit on the difference between germline and somatic editing and why this could be so scary.
100%.
All right, so stay tuned. Hi, this is the disembodied voice of Adam. I hope you're enjoying the show. If so, please like and subscribe, and consider finding us on social media. You can find us on Twitter and Instagram at AlmaMAC_radio. You can find me, Adam, on Twitter at Adam, FORTAIS. And find Sean on Instagram at Sean Hercules. Older episodes of the AlmaMAC and other great shows can be found on all streaming services by searching Scientific Canada, or you can go to anchor.fm/SciCan. Thanks. Okay, we are back. This is the AlmaMAC. This is Adam. That's Sean.
Sean.
And we're talking about CRISPR babies.
CRISPR babies.
I think I want to change my social media handle to that.
Oh,
CRISPR baby.
That's a lot.
Yeah.
Why?
I don't know. Maybe that's not very sensitive. Maybe I shouldn't do that.
No.
So we, before our commercial break, mentioned germline and somatic editing. So the whole idea of these CRISPR-Cas9 genetic editing things is to go into the genome and pluck out bad stuff and put good stuff in. But genomes get passed down.
Yeah. So what he was trying to achieve was... So he was trying to alter the germline. So a little background information. So germline mutations or germline DNA are those that can be inherited, passed on from offspring to offspring. However, the somatic genetic background is particular to specific tissues, organs, or cells. So, for instance, CRISPR-Cas9 has been used for, or can be used, has a potential to be used for, and ethically, for somatic mutations. So, if there's a particular mutation in a particular gene after the tissue and the human has been developed, you can use this. And it's okay, because the person has already been developed. You've done all of your research for what are the off-target effects and what's not.
Provided the patient understands everything that's going to happen, and they consent to it, then whatever happens to them stays with them.
And we'll put in the show notes examples of somatic germline, of CRISPR somatic editing. We'll put that in the show notes to give you specific examples. However, the germline is that you're altering the part of your DNA, like the regions of your DNA before full development, and that can be passed on from offspring to offspring, which, these babies did not ask for that.
Yeah, this brings all sorts of weird ethical questions, like consent down the genetic line, humanity's consent, introducing this into, and then the whole idea of designing the human race's genetic pool. That seems messed up, for lack of a stronger word.
Yeah, there's so many ethical concerns. We actually can't do this all in this one show. I'm looking at the time what?
And this is why it takes so long to go through an ethics board test. And I guess that's why you wanted to skip it all.
Yeah, sometimes it's this is a lot of work, but it is so necessary. So while you're waiting for ethics, you just do other experiments, you don't need ethics for. But yeah, it takes forever to go through this ethical review process.
I kind of fell down the rabbit hole of nightmare scenarios of germline editing. It seems like it's perfect for sci-fi. I'm sure there are 100,000 sci-fi stories about this. But worst case scenario, you end the human race. Best case scenario,
or you create this weird super race, which is so it seems too close to eugenics to me.
I think we can all agree that we don't want any of that nonsense happening.
And the thing about it is, there's so many things. But one of the things about it is that we don't fully understand what one particular gene or the effects of one particular gene or one particular mutation in a particular gene can have, because there's so many studies now showing that it's an interaction of particular mutations and different genes across the 23 chromosomes of the body, and a combination of those particular mutations that can lead to particular traits. Most traits are not monogenic, which means one particular mutation in the gene will lead to disease. Though there's a huge community of people living with monogenic diseases where CRISPR-Cas9 will 100% be beneficial, or germline editing could be beneficial. But we need to understand more before we can just jump and do this. Most diseases, most traits are polygenic, which means that they're a bunch of mutations and a bunch of genes that contribute to disease. And also you have to account for environmental factors. And yeah, this was just not the best representation of science for this particular technique.
This almost reminds me of, I don't know if this is true, I should fact check this, but I think I read somewhere or heard somewhere that, Mendel, the famous guy with the pea plants.
Peas, yeah.
If you went through high school biology and stuff, you've probably seen these Mendel genetic charts and stuff. I heard that he just fabricated all of that data. I mean, like, in essence, it's correct to some extent, but it's way, way more complicated, and it's like a nice training tool. If all of genetics was that simple, you just have this chart. If this is true, then this. If this is false, then this. Then maybe it would be a little safer, but that's just not the case. And
even then, even if you go with the whole Mendelian genetics kind of, for particular diseases that do follow that, there's still probabilities, right? So there's still a probability that the offspring will have this particular trait, given that they have this particular background. So it's so complicated, it's so complex that you can't just do that. I understand, though, from the scientific background that you want to be the first. You want to be like the superhero. So you're just gonna
this guy will be in history textbooks probably forever.
100%.
I mean, as a villain,
as
far as I can tell,
kind of. It depends on how you look at it, I think. There are a lot of people in his camp, actually. A lot of people not in his camp as well. It's just a huge ethical soup of opinions right now. But I personally am in the camp that is not.
Yeah, I feel like it's cut and dry for me.
Yeah.
So to sort of wrap up, we talked a lot about gene editing and the two different types, somatic and germline. Maybe I can get your opinion before we go. Do you think it'll ever be ethically sound to do germline edits, or do you think it's just way too complicated? Obviously, you don't have a time machine. Well, I mean, I don't think you do, but
I might. I wish I did.
Somatic for sure. That makes a lot of sense. It seems safe.
I think it could be extremely beneficial for. So anytime you do IVF, you have to have a screen done.
And that's in vitro fertilization.
In vitro fertilization, sorry. There's some kind of genetic screening done to assess the genetic background of the sperm and the egg, essentially. And if... Yeah, it's kind of tricky. But if there's a strong disposition to a particular monogenic trait that could harm the child or the offspring, I believe it could be extremely beneficial for the parents to have that discussion and either manipulate the DNA or not. Because there's also a chance that the offspring will not have that particular trait, right?
Let's see. I'm trying to think of a specific example. Can you think of any one off the top of your head that's like a prime candidate for this?
Like there's certain types of cancers. There are some developmental disorders as well. But we'll put that in the show notes as well.
Yeah, I feel like there's maybe a short list that's available readily of like prime candidates that are, monogenic, potentially not messy, pretty easy ones to go in and nip in the bud, so to speak.
Yeah, so there's this thing called pre-implantation genetic testing, which is what I was just alluding to. And yeah, so that will give like a screen of which particular traits potentially the offspring could have. But yeah, I don't know. It's also weird talking about this future baby as offspring because like they're going to, they're going to have their own
that's like an
autonomy
ethical bit that we just barely touched on that could be podcast after podcast of like, what if the offspring was fine the way they were and wouldn't have wanted this to be done?
Yeah.
I guess there's all sorts of things like that. Less intense, like I guess that circumcision is kind of like that. But that's reversible to some extent.
What? Circumcision is reversible?
I've heard of it.
I've never heard of that. But
anyway, this is a lot more serious than that. I don't know. If we were to vote, 2 out of 2, send this guy to jail. 3 years, long enough?
No. Well, actually, I don't know the law that well enough to actually say whether 3 years is long enough. It depends on what he was actually charged for.
Yeah, so my understanding, it was basically just like tampering with documents and medical malpractice, like a lower grade version of that. I guess because like these two offspring are alive. They seem to be living a normal life so far.
We don't know.
But we have no idea. They could have...
We don't know.
Akira-like superpowers.
Or not. Or could have... Or we have no idea.
Could have went in the opposite direction.
Yeah. And yeah, it breaks my heart actually, because we probably will never know the outcome of these two babies.
You'd want to keep their identities hidden for sure.
Yeah. I guess, I don't know, in 20 years or something, we might know, but we don't know right now. We probably will never know. And yeah, this was not a good display for science at all.
It's nice that a lot of people are rallying behind condemning this.
Yeah.
Especially the country of origin, where I feel like China's not getting a lot of great press these days with all the various conflicts that are happening. But this is a very strong show of ethics from them to prosecute and everything. Yeah.
But yeah, in short, we just need to do science better. We need to be more considerate of working with humans or like any type of organism. Anything we're doing in the lab, we should really take note of why we're doing what we're doing and just be more compassionate, because I can't imagine working in a lab that was doing that.
Like I feel like the parents were tricked into doing it under the guise of potentially helping their child. I think the father was HIV positive and didn't want his offspring to grow up like that, which is noble for sure. But without the right information, you're gonna you get duped. Yeah. Anyway, thanks a lot, Sean, for being the expert today.
Expert.
And here comes Get Lit. Stay tuned.